Hello learners, I am Dr. Rekha Sharma, former professor, UGC Human Resource Development Center, Raष्ट्रसंत तुकडोजी महाराज Nagpur University, Nagpur. In this module, we shall study malting of grains. Malting is a controlled germination process which activates the enzymes of the resting grain, resulting in the conversion of cereal starch to fermentable sugars, partial hydrolysis of cereal proteins and other macromolecules. Barley is the grain most generally used in the production of malt, which finds industrial uses in breweries and distilleries. In commerce, the term malt is applied to barley malt. Small quantities of other cereals are used for malting and they are designated as wheat malt, ragi malt and jowar malt. Now malting process. It starts with pre-cleaning, cleaning, grading and storage. Then the next step is steeping, followed by germination, kilning and separation of culms. Now let us start with the process. The first process is pre-cleaning, cleaning, grading and storage. Now after harvesting, barley is prepared for storage during its period of dormancy before conversion to malt under conditions minimizing the risk of infestation from insects, fungal attack, etc. To achieve this, it is necessary to dry the wet or green grain from the fields for long-term storage down to a moisture content of 12%. Grain with moisture levels between 10 and 12% can be stored for long periods, although initial drying down to 15% to 16% could be applicable, providing that the grain is stored for short periods and preferably cooled. Cleaning has to remove impurities of about the same size as barley, such as weed seeds of grains of other cereals as well as broken grains or undersized grains. Now steeping. Steeping initiates malting and is a crucial step in producing quality malt. During steeping, barley takes up water and swells by 1/3. Respiration increases slowly at first and then more rapidly, causing the grain in steep to accumulate carbon dioxide and heat up. This is aggravated by the action of microorganisms associated with the barley. The steeping stage consists of alternating periods when the grain is immersed in water, referred to as the underwater period, and periods with the water drained from the grain, referred to as the air rest periods. This combination is required to promote and maintain the efficiency of the germination. To achieve satisfactory results during the subsequent malting, it is necessary to increase the water content of barley to approximately 42 to 46% during the steeping process in a typical temperature range from 15 to 20 degrees centigrade. When barley absorbs water, the embryo becomes active and uses the oxygen dissolved in the steeping water for respiratory purposes. During the first period under water, the dissolved oxygen in the water is rapidly adsorbed by the organic material and microflora in the grain. In addition to supplying water and oxygen to the barley, steeping is of considerable value in removing dirt from the grain. The film of dust and many of the microorganisms adhering to the grain are washed away by the steep water, and this cleaning action can be increased if special equipment is used to agitate the barley during steeping. Steep water leaches husk components that may impede germination and any musty smell that may be associated with the barley, and possibly some that prevent proper flocculation of yeast. Now, germination. The germination process is characterized by the growth of the embryo of the grain, manifested by the rootlets, growth and increase in length of the shoot, that is acrospire, with the concomitant modification of the contents of the endosperm. A large increase in the quantities of some of the hydrolytic enzymes present in the grain is one of the essential changes of modification. The alteration of the starch endosperm begins due to the partial degradation of reserve substances, cell walls, gums, proteins and starch. The hydrolytic enzymes that catalyze modification are derived mainly from the scutellum and aileron layer. In the initial stages of germination, the scutellum releases many hydrolytic enzymes that begin to degrade the cell walls of the crushed cell layer and the walls, proteins and starch granules of the endosperm. In addition to enzymes, the scutellum releases gibberellins which stimulate the production and release of numerous hydrolytic enzymes by the aileron layer. Alpha amylase is absent from the hydrated aileron and its de novo synthesis and release are independently triggered by the arrival of the hormone. Alpha amylase is absent from the hydrated aileron and its de novo synthesis and release are independently triggered by the arrival of the hormone. It is estimated that in malt, approximately 85% of this enzyme originates from the aileron layer and 15% from the scutellum layer. Beta amylase, which exists in the original barley in an inactive form in the starchy endosperm, is liberated and the degradation of the components proceeds. Proteases and carboxypeptidases accumulate in the starchy endosperm during germination. Beta-glucan solubilase is a carboxypeptidase that converts the insoluble cell wall protein matrix to high molecular weight soluble but viscous beta-glucans, which can create problems during wort separation and beer filtration. Those are broken down further to low molecular weight glucans and glucose by the endo-beta glucanases and beta glucosidases. These enzymes develop slowly in the malting process and are slightly sensitive to gibberellic acid. Gibberellic acid is an example of germination regulator which, like potassium bromate, can be added to the grain to greatly accelerate the modification rate. At this stage, the material is called green malt. Now, kilning has the characteristics of a typical industrial drying process occurring in two distinct stages. First, moisture is removed from the green malt, reducing it from approximately 44 to 12%. This phase of drying is rapid and is referred to as the free drying or withering phase. The second phase of drying, in which the malt is dried from 12 to 4%, occurs in a much slower process which is commonly referred to as falling rate phase. At the end of the drying process, the kiln temperatures may be raised for 1 to 2 hours in the curing stage, followed by a cooling period to achieve a temperature suitable for discharge and storage. Kilning not only dries the malt, preventing the further growth and modification, but also removes undesirable flavors, partially or wholly destroys some hydrolytic and other enzymes, and develops flavor and color in final product. Moreover, malt is kilned to produce a friable, readily milled, stable product that may be stored for long periods and from which roots may easily be removed. Now, separation of culms. When kilning is complete, the heat is switched off and the grain is allowed to cool before it is stripped from the kiln in a stream of air at ambient temperatures. On its way to steel or concrete hopper bottomed storage silos, the malt is dredged to remove dried rootlets or culms, which are extremely hygroscopic and must be stored out of contact with the air. They have a much lower economic value than the malt and are used for feeding cattle. Approximately 3 to 5% of the original barley is recovered as culms in traditional malting process. Now, uses of malt. Approximately 96% of the malt produced in the world is used as main raw material along with water to make beer. Likewise, malt is extremely important in Scotland for whisky production, but this only represents approximately 3% of world's malt use. The rest is used as a very important ingredient in the food industry as natural flavoring, breakfast cereals, malted milk drinks or malt vinegar. Now, let us summarize. Malting is a controlled germination process which activates the enzymes of the resting grain resulting in the conversion of cereal starch to fermentable sugars, partial hydrolysis of cereal proteins and other macromolecules. The malting operation starts with the drying of the grain in a kiln so that moisture content of the grain is between 10 to 14%. After drying and cleaning, the grain is stored for at least 3 weeks before malting. For malting, the stored grain is steeped in water for some 50 to 70 hours, after which time the surplus water is drained off from the grain, which is then sprayed on the floor for 7 to 8 days while germination takes place. The grain malt is then kiln dried to arrest enzymatic activity without destroying the enzyme. The dried and cleaned product is malt. These are references. Thank you so much.